Hello everybody. Today we'll talk about the cell, the structural and functional unit of the living organism. All the living organisms composed of cells. However, some living organisms are composed of hundreds of billions of cells. For example, human body. Many different types of cells are together to form the human body. There are some organisms, those are called microorganisms. They are composed of only a single cell, unicellular. For example, bacteria or amoeba. So those are microorganisms. A single cell is a single body. Okay. However, cell is the smallest unit, structural and functional unit of the all living organism. First, we'll talk about the structure of a cell. A typical cell consists of a boundary, that is the cell membrane, and inside the cell you will find a fluid part that is called the cytoplasm and a round structure that is the nucleus. So we'll see the structure and functions of the cell membrane. Then we'll talk about the proteins present in the cell membrane and their functions. There are two types of proteins and they perform some important functions. We'll talk about that. Then we'll talk about the junctions that attach the cells or help to communicate between the cells. Those are called intercellular junctions. Then we'll talk about the transport across the cell membrane. Cell membrane is a semi-permeable membrane. So how the molecules cross the cell membrane, that semi-permeable membrane, there are different methods or ways of transport of the molecules across the cell membrane. So we'll talk about that. Then we'll see the response of a living cell in three different types of solution: isotonic solution, hypertonic solution, and hypotonic solution. How the cell responds in those three types of solution. Then we'll talk about so those are the things we'll talk today. So first, it is important to know that there are 50 to 100 trillion cells in the human body. Not million, not billion, 50 to 100 trillion. And over 200 different types of cells are present in the human body, and they are different in terms of size, shape, their structure, functions. So over 200 different types of cells we have in the human body. However, one thing is common: all cells are composed of mainly four elements: carbon, hydrogen, nitrogen, oxygen, CHON or CHNO, form most part of a cell. As I said that over 200 different types of cells are present in the human body. How they are different? They are different in shape, size, in their functions and morphology. If you see the color, they are different from each other. Here you see few examples of different types of cells. Although we have more than 200 different types. Here you can see few examples how they could be different from each other. So first, top left, here these cells, these two cells are fibroblasts, connective tissue cells. Most common type of connective tissue cells are fibroblasts, and they produce fibers. So you see these are the fibers, so fibroblasts. These two are the red blood cells. They are round but flat, and the color is red, and the function is oxygen transportation. Red blood cells are also called the erythrocytes. Same thing, okay? RBC, or red blood cell, or erythrocyte. So you can see that they are different from each other in terms of shape, function, right? Color. Red blood cells look red because of the hemoglobin inside. These 5 cells are epithelial cells, okay? And epithelial cells usually cover the surface or inner layer of the organ. And now you see this is a skeletal muscle cell. So the cell is pretty big, long. Usually skeletal muscle cells are very long and many nuclei inside. So you can see inside one cell you have multiple nuclei. These are smooth muscle cells, and smooth muscle cells are smaller, much smaller than skeletal, and both ends are pointed, you see, and a single nucleus here. So even different types of muscle cells are different in terms of shape, size, and other features. And the muscle fibers or muscle cells are mainly responsible for the movement of the body parts or movement of the bones, right, by their contraction. So function is different. This is a fat cell. You see something interesting here. The nucleus is not in the center but attached to the membrane, and the function of the fat cells is storage of energy. So fat cells store energy, the source of energy. This is a macrophage. It's a phagocytic cells, do phagocytosis. The term phagocytosis means engulf. Okay, so this cell has multiple processes, and macrophages do what? Phagocytosis. They engulf the dead cells or injured cells or microorganisms. Okay, so cleaning and providing defense are the important functions. Nerve cell. You see the nerve cell has a cell body, and inside the cell body this is the nucleus and multiple processes. This long process is called the axon, and these short processes are called the dendrites. Okay, so you see that a nerve cell, which is also known as a neuron, is different than other cells, right? And the main function is electrical signal transmission, transmits electrical signal. The last one here, this is a sperm. Sperm is a male sex cell, and sperm has a head, midpiece, and a tail, and it can swim, and nucleus is located inside the head. So you see it looks like a tiny fish, right? So the function is reproduction. Sperm fertilizes the egg or ovum, and that produces the new life, right? So you see a few examples how different cells in the body could be different in terms of their shape, their size, their color and functions. Okay, now we'll see the parts of a living cell. If you see a typical cell, as I mentioned before, it has a cell membrane or plasma membrane. So this is the plasma membrane that gives the boundary of the cell and protects the cell, gives the shape of the cell. So those are three functions of the plasma membrane: gives the boundary, protects the cell, and gives the shape of the cell. And the plasma membrane separates extracellular fluid outside of the cell, the fluid, extracellular fluid, and intracellular fluid inside the cell. Okay, so those are the important functions of the cell membrane. Now, cell membrane is a semi-permeable membrane. That means it is not fully permeable, partially permeable. Some small molecules can pass through it, but not everything. Okay, then usually in the center of the cell there is a nucleus, okay, which is called the control center of the cell. And in between the plasma membrane and the nucleus, the fluid part is called the cytoplasm. Okay, so this is the cytoplasm. Now, in the cytoplasm, you have organelles, okay, liquid and some organelles. There are different types of organelles. We'll talk about that. Okay, so cytoplasm has liquid and the organelles, and organelles could be two types: membranous and non-membranous organelles. Okay, so some organelles have membranes around them, bounded by membranes, and some organelles don't have membranes. So we'll see the important organelles in next slide. So these are the three parts of the cell: the plasma membrane, nucleus, and in between you have the cytoplasm, and cytoplasm has membranous and non-membranous organelles. Okay, important organelles are, let's see, this is a nice picture of a typical cell. So this is the plasma membrane or cell membrane, this is the nucleus, okay, and in between in the cytoplasm you have the organelles. You see different colors. So this is called endoplasmic reticulum or ER, and endoplasmic reticulum are tube-like structures, many tube-like structures together. They are connected together to form the endoplasmic reticulum around the nucleus. So these blue structures are the endoplasmic reticulum. Okay. Now, endoplasmic reticulum has two parts. You see, if you see this part, you see on the surface you have tiny structures. These are called the ribosomes, the tiny red dots. These are the ribosomes. And the part of the endoplasmic reticulum that is covered by the ribosome, or that has ribosomes on the surface, that is called the rough ER or rough endoplasmic reticulum. Okay, here. And this part you see no ribosomes on the surface of the endoplasmic reticulum, and that's why this part is called the smooth endoplasmic reticulum or a smooth surfaced endoplasmic reticulum. So two parts, rough because the ribosomes are attached on their surface, smooth, no ribosomes attached on the surface. Okay. Now ribosomes, these tiny structures, do what? The ribosomes are very important because they produce or manufacture the proteins. Okay, so ribosomes do what? Protein manufacture, synthesis of protein. Okay, and then what happens? The protein molecules enter into the rough ear because ribosomes are just attached on the outer surface of the rough ear. So after the protein molecules are manufactured, they will enter into the rough ear. And rough ear do what? Rough ear will process the proteins, okay, that it receives from the ribosomes. Processing means folding, we can say folding of the proteins, cutting of the proteins, okay, giving shape, correct shape. So these are the processing of the proteins done inside the rough ear. Then the processed proteins go to these green structures here. These are called the Golgi Apparatus. Okay, they are like flattened membranous sacs piled together or stacked together, and both ends are expanded. So like this. Okay, so several flattened sacs are together, and both ends are expanded or wider, and the protein molecules, those are processed in the rough ear, they enter from rough ear into the Golgi apparatus. Okay, so Golgi apparatus. And the Golgi apparatus sends the protein molecules to the ends of the flattened sacs. So the protein molecules are accumulated at the ends, and then what happens? The end parts get detached from the main part, okay, of the Golgi apparatus. So these get detached, and these are the proteins. Okay, the proteins could be hormones or enzymes. Okay, digestive enzymes that can destroy the antigens or bacteria or virus, okay, or other hormones that should be secreted. Okay, and these round membranous sacs filled with the protein molecules are called now lysosomes, okay, or vesicles. Okay, so the vesicles or lysosomes, they get detached, you see here, and they are filled with the protein molecules. Okay, and then they will go to the cell membrane or plasma membrane and will rupture, and that process is called exocytosis. So these vesicles or lysosomes will go to the cell membrane and will get attached to the cell membrane and then rupture to release the protein molecules. It could be enzyme or hormone or other secretions, and that process is called exocytosis. Okay, so you see that if we just quickly review who produce or manufacture the proteins? Ribosomes. Who process the proteins? Rough ear. And who does the packaging of the protein? Golgi Apparatus. Okay, and send the vesicles to the cell membrane, where they rupture the vesicles to release the chemicals to the outside of the cell by exocytosis. Okay, the process. Okay, now smooth ear. Smooth ear performs different functions. A very important function that is called detoxification. Detoxify the toxic chemicals, neutralize the toxic chemicals, right? And another important function is lipid synthesis. Lipid synthesis. Okay, so that is the smooth ear. These structures, you see orange color, these are very important. These are called mitochondria, and mitochondria are known as power houses. Okay, so mitochondria are called the powerhouses. Why? Because mitochondria produce ATP, the source of energy. ATP, remember, very important chemical, that is called adenosine triphosphate or ATP, and it releases energy which our body always needs. That's why mitochondria are the powerhouses. Most of the ATPs are produced in the mitochondria. Okay. Also see the nucleus. This is the nucleus, right? And nucleus is covered by this membrane that is called nuclear envelope. Okay, like a cell. It is also covered. Nucleus is also covered by a membrane, and that is called the nuclear envelope. Okay, and in the nuclear envelope, you see there are many pores or channels or openings. These are called the nuclear pores. So these are pores in the nuclear membrane. So the chemicals can, get out from the nucleus. Now, inside the nucleus, this structure is called nucleolus. And nucleolus contains the DNA and RNA. Very important components of the cell. DNA and RNA are present in the nucleolus, densely present in the nucleolus. Okay, deoxyribonucleic acid or DNA and ribonucleic acid RNA. Now, DNA is responsible for the hereditary information that goes from the parent to the offspring, right? So DNA are very important components. And if you see inside the nucleolus, what you will find, chromosomes. Okay, thread-like structures, and in the chromosome you will find DNA. So DNA make the chromosomes, and Inside the chromosomes, you will find the gene, okay, and the gene is formed by the DNA. So chromosomes, DNA, and genes are present in the nucleolus, and they are mainly responsible for hereditary information passed from the parents to the offspring. Okay, very important. Now, RNA are mainly responsible for the synthesis of a part of the ribosome. You remember the ribosomes, these tiny dots, structures attached on the surface of rough ear, and they produce or manufacture the protein, right? Very important function. So important part of those ribosomes are produced inside the nucleolus, and that are the RNA. So RNA gets out through the nuclear pore. So produced in the nucleolus, RNA, and get out through the nuclear pore and form the ribosomes, and those ribosomes get attached to the endoplasmic reticulum surface. Okay, and in between the nucleolus and the nuclear envelope, this part is called the nucleoplasm. Okay, nucleoplasm, also known as chromatin. Nucleoplasm mainly contains fluid, a lot of water, and ions and other chemicals. Okay, so uh, those are some important structures and their functions in a typical cell. You see these, and these are the centrosomes. Centrosomes are responsible for cell division, participate in cell division, and also inside the cell we have microtubules, microtubules and microfilaments. They are mainly responsible for giving the shape of the cell. So these are also known as cytoskeleton. Cyto means cell, skeleton. Microtubules and microfilaments. These thread-like structures you see here. Okay. Now we'll see the microscopic structure of the plasma membrane. Plasma membrane is a phospholipid bilayer. That means what? Phospholipid molecules form two layers. Okay, so this is one phospholipid, this is another phospholipid. Another phospholipid here, this is another phospholipid. So the phospholipid units form two layers. Okay, so this is one layer of phospholipids, this is another layer. That's why it is called a phospholipid bilayer, and in that phospholipid bilayer you will see the protein structures. This is a protein. Some protein structures pass all through the phospholipid bilayer like this one, and this is called the integral protein, and some proteins are attached to the integral protein, and this is called the peripheral protein. Okay, so integral proteins are deeply inserted. Some of them pass through the phospholipid bilayer completely, some of them are deeply inserted. So these are integral proteins. Okay, and this is a peripheral protein. If you see another one attached here, this is a peripheral protein. peripheral proteins. Okay, so two types of proteins are in the cell membrane. Make sense? So if I ask you what is the structure of a cell membrane, you'll say it is a phospholipid bilayer in which the proteins are present. Okay, and how many types of proteins? Two types of proteins. Some are deeply inserted, those are called the integral proteins, and some are loosely attached to the integral proteins, those are called the peripheral proteins. Make sense? So now you see here, this is one layer of phospholipid, this is another layer of phospholipid, right? And this is integral protein, these are integral proteins, and this one, these are peripheral proteins. Okay, those are two main structures, phospholipid and proteins. Now also in the cell membrane you see cholesterols, glycolipids. That means you see these yellow structures or orange, these are cholesterols. Okay, these are cholesterols, and these are glucose. And sometimes you see that glucose and cholesterol are attached. So this whole structure will be called glucose and lipid cholesterol, glycolipid. Okay, and glycoproteins. Those proteins and glucose molecules joined together called combinedly called glycoproteins. Makes sense? Glucose and protein, glycoprotein, glucose and lipid or cholesterol, glycolipid. Okay, uh, now if you see a single phospholipid unit looks like this, right? And this is called the head of the phospholipid unit, and this is the tail part. Okay, and head part is water soluble, that means polar end, and tail part is water non-soluble, that means non-polar end. Okay, same way, this is another phospholipid, so this is the head polar, this is the tail non-polar. Now you see the middle portion from here to here, this part of the cell membrane doesn't have any head, only tail. That means water insoluble. So water cannot pass through this part, tail part. Okay, only head part is water soluble, and therefore the phospholipid bilayer doesn't let the water molecules pass through it. Makes sense? Because the tail parts don't like water, and that's why tail part is also called hydrophobic part. Phobia means fear, scared. Hydro means water, so water disliking part, and head part is polar part, that means likes water, and that's why it is called hydrophilic. Okay, hydrophilia, philia means attraction, so water liking part. Okay, so that is the structure of a cell membrane. Now there is a theory to indicate or explain the structure of the cell membrane, that is called the fluid mosaic theory. What is that? Fluid mosaic model. Why it is called a fluid mosaic model? Because in phospholipid bilayer, those protein structures continuously change their shape. So when you see under the microscope, very powerful electron microscopes, you see it looks like in the phospholipid layer, the proteins are moving. Okay, proteins are changing shape, moving. So it looks like fluid under the microscope. That's why it is called fluid mosaic model. Nice, beautiful stones in the phospholipid layer. Now, integral proteins in the cell membrane perform few important functions: transport by acting as channels. Some integral proteins work as receptors. Okay, and if you see some integral proteins, you will find through the integral protein, this is an integral protein, you have openings all through. So from outside, the small molecules including water can get in, some molecules can get out. So these are channel proteins. Not all integral proteins have channels. Some integral proteins have channels. So these are channel proteins. Okay. Some integral proteins, they have the binding site like this. So what will happen? A molecule that should be transported through the cell membrane will first sit here, and then this integral protein will change the shape and will take the molecule inwards like this, and eventually will take it inside. Okay, so by changing the shape, first the molecule will bind, then the integral protein will change the shape and then push it to the inside. So these are two main ways the integral proteins, certain integral proteins transport. Okay, by forming channel or by uh, moving the molecule by changing its own configuration. Okay, integral proteins own configuration. Now channels could be two different types. Some channels are always open. For example, this one, channel integral protein. So this channel is always open. However, there are some integral proteins that have the channels. Those are gated channels controlled by ATP energy. So the channel is usually closed, but when ATP works on it, it will get opened. So the gated proteins are controlled by the energy that comes from the ATP. However, this channel doesn't change; it always open. But this one opens and closed, gated. Okay, so that's how the transport proteins work. You see here, this one is changing configuration. What happens? You see, this is interesting. First, this protein will come close to the integral protein, and there is a binding site here, so it will sit here like this that I mentioned before, and then this gate will get closed, and this side will get open. Okay, and it will get detached and will get out. Okay, or get in. So by changing the shape of the protein, some integral proteins work as the receptors. What happen? You see, let me give you an example. This is one integral protein, and this one has a binding site which is triangular. This is another integral protein. This one has a round binding site, and this is another integral protein. This one has a square or rectangular binding site. Okay, so these are three integral proteins. They are working as receptors, and this is outside of the cell and this is inside of the cell. This is the cell membrane, right? So what happens, these receptor integral proteins have binding sites at the outer pole, and the binding sites have different shapes. Now if a hormone is released and that will work on this cell membrane of this cell, what happens, if the hormone molecule, the shape of the molecule is round, then what will happen, it will bind perfectly here, right? Because this is binding site is round. Okay, and when that binding will be perfect fit, it's like key lock type, binding, then a signal will go to inside the cell, from outside to inside. Okay, so this hormone molecule or the medicine you take that works on the cells of the body, these are called the signal molecules. Signal molecules bind with the receptor proteins. Okay, and when the binding is perfect, signal will go inside and intercellular machineries will start to work. Okay, it's like, you put your key in the car and turn, and that starts the engine. Same way, if this one goes there, it will say no, my binding site is not perfect for you. Goes here, it will say no. Now if the medicine molecule or hormone molecule, that means the signal molecule, is rectangular, then what will happen, it will perfectly fit here, right? So this is the signal receptor binding, and when that binding will be perfect, this one will send signal to the inside the cell. Okay, if the signal molecule is triangular, it will bind perfectly here. So that's how different integral proteins work as receptors, and when the binding occurs, signal goes to the cell, inside the cell, and the DNA and other RNA, they will start to work, and that is how the medicines or hormones work on the cells, activate the cells. Okay, so some integral proteins present in the cell membrane work as the receptors. Some integral proteins act as enzymes. That means change one chemical to another, one type of chemical molecules to another type. For example, this one is working as enzyme 1, this one enzyme 2. So you see, when this molecule comes contact to this one, it will be changed to this. So this will be changed to this by this one, and then this integral protein will work as another type of enzyme that will convert this one to this one. Okay, so those are three important functions of the integral proteins. Now we'll see the important functions of the peripheral proteins. Peripheral proteins perform two very important functions. There are other functions, but these two are very important. Intercellular joining or junctions between the cells. So this is one cell, this is next cell. You see in many different tissues, the cells are attached to each other by the peripheral proteins, because the peripheral proteins form the junctions or joinings. Those are called intercellular junctions or joinings. Okay, so they attach the cell members adjacent cells to each other. Okay, very important. So the cells will not get dispersed or will not get separated from each other. Also, some peripheral proteins have channels, through channels, or you know openings or pores, so the chemicals can go from one cell to the next cell. So holding them together, as well as some peripheral proteins have channels, so the fluid can go from one cell to another. Cell to cell recognition. Uh, what happens in some cases, the peripheral proteins, like this one is a peripheral protein, this is a cell membrane, this is integral protein, this is peripheral protein. Okay, phospholipid bilayer. These peripheral proteins can detect the next cell or adjacent cell, and that is called cell recognition, cell to cell recognition. So when, for example, this is one cell membrane in your body, now if you get another cell from a different person's body, this peripheral protein will be able to detect that it is not from your own body but coming from outside. And after the peripheral proteins detect the cell, then it can accept or reject. In many cases, most of the cases, your body doesn't accept another person's tissue or cell, because your cells can detect others' cells, right? Can detect if the cells are from your own body or from outside. That's a big challenge, right? The tissue rejection or organ rejection when transplantation of tissue or organ is done. That's a big problem. So the peripheral proteins can detect the adjacent cells. Now, I said you that some peripheral proteins form junctions or joinings. Three types of junctions or joinings are the most common types: tight junctions, desmosomes, and gap junctions. Okay, these junctions mainly uh, hold the cells together. Okay, so the cells will stay together. That is important. For example, when muscle contraction occurs, right, the cells must work together at the same time. So also when the heart contracts, the heart muscle fibers or cells all should stay attached to each other strongly. Makes sense? Gap junctions have pores or channels, so the ions or molecules can move from one cell to the next. Tight junctions allow very small molecules only pass through it, prevent fluids and most molecules. So most of the molecules are not allowed or cannot pass through the tight junctions. Only few and small, very small molecules. Desmosomes are to anchor the adjacent cells very strongly, like spot welding. So desmosomes actually uh, come out from the cell membrane and they hold each other very strongly, like zipper in your clothes. Very strong, right? The zippers you use in the clothes or in your pants. So desmosomes anchor or hold the cell membranes, adjacent cell membranes very strongly, tightly. Here you can see all three types of junctions or joinings have been shown. This one here, tight junction. So you see the cell membrane here and here, these two cell membranes. So this is one cell membrane, this is another cell membrane. of two cells, and these protein structures are the peripheral proteins form the tight junction, and these proteins actually stretch the cell membranes together, like when you put two clothes together and stitch them. Similar way, these peripheral proteins are attaching them together. Okay, and these are the tight junctions. Now next, the desmosome. You see, this is one cell membrane, this is another cell membrane, this is a desmosome. So if you see the structure, you see this is the cell membrane of one cell, this is the cell membrane of another cell, and in the inner surface of the cell membrane you have the plaques, disc like structures. Okay, and from the plaques these protein structures come out, and these are called linker glycoproteins. So these are proteins, glycoproteins, called the linker glycoproteins or cadherins. Important. Okay, I may ask you. So the cadherins or linker glycoproteins come out from the plaques and get attached to each other like this. This is one linker glycoprotein, this is another linker glycoprotein. So they get attached to each other like this. So very strong, like zipper. This is a gap junction here. This is another. So as I mentioned, through the gap junctions you have the pores or channels. So here you can see better. So this is the gap junction and pore or channels. So ions can quickly go from one cell to adjacent cells. Okay, and they can talk to each other. Cells can quickly communicate or talk to each other. Plenty of gap junctions are present in between the cardiac muscle cells. So when changes occur in one area, it quickly spreads to the other cells. So the heart can contract together. Transport across the cell membrane. Through the cell membrane, as I mentioned, cell membrane is a semi-permeable membrane, right? So it is partially permeable or selectively permeable. It will not allow everything, but will allow some chemicals pass through it. Now, through the cell membrane, two ways the chemicals pass or cross: passive and active. The main difference is, for active processes you need ATP, energy. Okay, for passive no ATP is required. Okay, and passive is towards the concentration gradient, and active is opposite, against the concentration gradient. That's why you need energy, right? Like if you want to cross a river, if the current is in your favor, then you don't need to use energy, right? The waves will take you, the current will take you from one side to another side. That is passive. Now if you want to go against the wave or against the current, then you have to start the engine. You need to use energy. Okay, that is active. Now passive processes include diffusion, and osmosis. These are two main types. Okay, diffusion and osmosis. And uh, active is uh, against the concentration gradient that uses ATP. Now the differences between the diffusion and osmosis is, if this is the cell membrane, okay, this is the cell membrane, and you have solutions. This is solution A and this is solution B in the other side. Okay, both are glucose solution. Now the concentration of glucose solution in this side, side A is high, right? So this is high concentration and this is low concentration. So in this side you have more water, right? Amount of water is more. That's why it is low concentration, right? But solute, these are glucose molecules, these tiny dots. Glucose or solute is less. That's why it is low concentration. And this one is high concentration because water is less, but glucose or solute is more. So glucose is more in this side, right? And water is less. So now if I ask you very simple question, to bring equal concentration in both sides, which one should move to which side? Definitely glucose is more in this side in high concentration, so the glucose molecules will move from this side to low side, right? And that is simple diffusion. Is it clear? So the solutes move from the high concentration to the low concentration, and that is simple diffusion. Very simple. Okay, and always remember the water or solvent will move from the low to high, because in this side water is more, right? So water will move from this side to this side, and that is called osmosis. Okay, so the movement of solute from high to low is called the diffusion, and the movement at the same time of solvent or water from low to high is called osmosis, and both happen simultaneously to bring concentration equal on both sides. Okay, solute is more in this side, will move from here to here. Water is more in this side, will move from here to here. Okay, so very simple. Sometimes, so this is the simple diffusion. Now there is another type of diffusion that is called facilitated diffusion. What happens? These molecules will move from this high side to this low side. Okay, and sometimes it is helped by another molecule, like this molecule act as a carrier. Okay, so what will happen? This molecule will sit on this molecule on the shoulder of this molecule, and this will carry it to the other side, and that is the facilitated diffusion. Okay, it is like, if you want to cross a lake, you can cross by yourself, or you can sit on a boat and that can take you to the other side. Okay, either way you can go to the other side. So if you take sit on another vehicle, that is carrier mediated diffusion. Okay, but no energy is required in diffusion because it belongs to passive process. Okay, here this is a simple experiment that I showed you already, actually, in an U-shaped test tube in the lab. If you take a high concentration solution in one side and low concentration, this is a membrane you put in between. That membrane has large holes or pores. So what will happen? Both simple diffusion and osmosis will occur, right? Solutes will move from this side to this side, water will move from this side to this side. Okay, you see here. To bring the concentration, the goal, remember always, the goal is what? Bringing the balance, concentration equal on both sides. So very simple, if I ask you to bring concentration equal in both sides, which one should move from this side to this side? The solute, because you have more solutes here. Which should move from this side to this side? The water, because you have less water here. So finally the concentration will be equal on the both sides, in the both sides. Now if you put the semi-permeable membrane that has small holes, smaller than the solutes, in this case what will happen? Only water will move from this side to this side. Water molecules are small, right? But solutes will not be able to move. So the volume of water will increase because the water is moving until the concentration becomes equal in both sides. Remember, volume and concentration, two different things. Here the volume in both sides equal, right? But the concentration is high in this side, low in this side. Now here, although the volume in this side is more, but concentration in both sides equal. Okay, the ratio of the solute and solvent is equal in both sides. The volume is different. Response of a living cell in three different types of solution. Okay, now this is a beaker. I took some solution. Okay, and this solution, the concentration is 5 milliosmol. Milliosmol is the unit of concentration. So if I put a red blood cell, for example, here, and inside the cell, this red blood cell, the concentration of cytoplasm or fluid is also 5 milliosmol. So I'll say this solution, since the concentration of the solution is same, this solution is iso means same tonic, same concentration. So no need, right, of simple diffusion or osmosis because already balance is here. So nothing will get in or out. So the shape of the cell, red blood cell, will not change. So in isotonic solution, the shape will not change. Now if I put the red blood cell in a solution, this solution, the concentration is 10 milliosmol. Okay, the solution, and this is the red blood cell, which has always same, right? Let's say 5 milliosmol inside the red blood cell. Now which way the water will move? Water always moves from low concentration to high concentration, right? That is the osmosis. So in this case, since the concentration outside of the cell in the solution is high, the water will get out from low to high and the cell will shrink. So in this case, the cell size will get smaller. The cell will shrink. Makes sense, right? Because water will get out. Now if the solution here, the concentration of solution is very low, like 2 milliosmol, and the red blood cell inside is 5 always. Okay, this is called hypotonic because hypo means less, right? compared to this one, this solution is hypotonic. This solution is 10 compared to this red blood cell. This is hypertonic. Makes sense? More. So in this case, always the rule is same. Water will move from low to high. So water will move from the solution to the red blood cell, into the red blood cell. So what will happen? The red blood cell will get bigger, swelling, or even may burst. Swelling of the cell, and if it continues, the rupture can also occur, right? So you see that in isotonic solution, no change. In hypertonic, the cell will shrink because the water will get out. In hypotonic, water will get in from low to high, so the cell will get bigger, enlarge, and may rupture. Okay. So when you will be asked to give the fluid or saline into a patient's body, you will prefer this one, right? That will not bother the cell. So here you see isotonic, no change, hypertonic, shrink, right? And hypotonic, the cell will get bigger. So those are the things you need to know from the cell, the important things that I went over. The different types of cells in the body. We have more than 200 different types of cells. I gave you a few examples. You need to know those cells. I talked about their shape, functions, and other things. Then I talked about the structure of a typical cell. Cell has a plasma membrane, its function, structure. Then I talked about the nucleus, right? And in the nucleus, you have the nuclear envelope, nuclear pore, you have nucleoplasm and the nucleolus, and the important functions of the nucleolus. Then I talked about the organelles, mitochondria, endoplasmic reticulum, ribosomes, right? Golgi apparatus, lysosome, centriole, right? Those are the important organelles. Microtubules, microfilaments. Then I talked about uh, the structure of the cell membrane, right? Proteins in the cell membrane, two types of proteins and their functions. Then I talked about the transport across the cell membrane, intercellular junctions or joints formed by the peripheral proteins, three types of joints or junctions I talked about, right? Tight junctions, desmosomes, gap junctions, and the response of a living cell in three types of solution: isotonic, hypertonic, hypotonic. So those are the things you need to know. Okay, thank you.